Yeah, quick bit of coffee.
Yeah.
Always a good way to start the show.
Totally.
Dr. Anna Brooks. Yeah. Hey, Dr. Anna Brooks, thank you very much for coming out, making the trip out here today to the show. How are you?
Great, thanks. Lovely day out there.
Finally, here in New Zealand, we're getting summer and autumn.
I know, right?
Yeah.
We've had a rough ride up here, haven't we?
We have. This has got to be the wettest summer. I've been here for 20 years. I think it's the wettest I can remember it being. Yeah.
Definitely.
And I mean, if it weren't for the actual damage and real trauma that people have suffered, it would have kind of been all right, because this summer I didn't have to water the garden because there was heaps of rain, whereas normally we do. But, I mean, people not far from here, Muriwai, have just been...
It's been devastating.
Yeah. Yeah. Apparently a third of the homes over there are red stickered. People can't go back to their homes.
So awful.
Yeah.
So
it's not
awful.
cool at all. So today, can we talk a little bit about the immune system?
We can.
Yeah?
Yeah.
Because this is, I believe, this is sort of your background. How did you get into studying immunology?
Yeah, well, I never really planned my career in any way, shape or form.
does?
I know, right? But, even when I was at school, I was like, what am I going to do? Yeah. And interestingly, I did both sort of commerce and science, still not knowing what on earth I wanted to do. So I liked the accounting and all those bits and pieces as well, which was confusing. Yeah. And so off I went to university and did both degrees. And it was when I was at university in those sort of first years where I started to realise that the way that the things that intrigued me were the things I never knew. Rather than sort of learning about business in a, this is business and labelling it, it was what sort of really drew me in or sort of captured my attention was the learning new stuff and how things worked and everything. And even then I still didn't really know where I was headed. So I went through university bumbling along going, this is cool, and I keep following along. So I first studied down in Wellington at Vic Uni, and then sort of came to the end of my degree, did honours in kind of an immunology space. Interestingly, it seemed like, I won't say a new area, but almost. There weren't many sort of specialists around at the time.
When was that? When were you studying that?
So that was early 2000s. I always forget exactly.
Don't want to give away too much information.
No, exactly.
So that's okay.
A long time ago. That's what sort of really initially sparked my interest. And then I was ready to move cities. So I looked to Auckland, came up here. And again, I didn't sort of deliberately go into immunology. It was just, things fell into place. And so I embarked on an immunology PhD. And the sort of the core focus of my work back then was understanding, when we take blood out of humans, and start to understand, what drives them and what keeps them alive for sort of essentially what we were my PhD was around was how do we keep cells alive in the laboratory?
Great question. I mean, just in general, how do they stay alive?
Yeah, exactly. And therefore, what drives them and, all the bits and pieces and all of that sort of complicated stuff so that we can understand them further. Like I said, obviously there's loads that we want to understand about the immune system and immune responses, but my question was how do we, nurture them
in the lab
in
so
the
we
laboratory
can study
setting.
them.
Exactly. So that's kind of where I started. Yeah. And so, that sort of started my path. And so complicated.
Let's go back to that. So what did you find? Did you find techniques to be able to increase cell life in the lab, or what were you trying to determine there?
What we wanted to determine is the particular cell type that I was looking at are our T cells. So with the immune response, we have the first line of defense that sort of kicks off like a sprinkler system, and we call that non-specific. That's, so it kicks into gear immediately when a foreign, virus, bacteria or anything enters our body, whether that's inhaled through the skin, all those sorts of things. So we have first line of defense, and what we were looking at is the second line of defense, or what we call a more specific immune response, where it's tailored. So
it's experienced something before it's developed specific antibodies for that.
Well, exactly. In fact, it's what I was specifically looking at was the naive cells, and they're literally called naive T cells or naive B cells, where we have a repertoire of cells floating around our bodies, and they're waiting for that moment that they're the one that needs to see this new foreign thing. And, we can talk about COVID in that sense as
Oh,
well,
yeah.
because that was a new foreign virus that the body had not seen. So what we want to do is we hope that our naive cells that were, swimming around, circulating or patrolling our bodies looking for a foreign threat, sees it and goes, oh, here we go, and starts to get triggered. And then essentially what happens with T cells then get triggered to clone themselves into a big army, right? So we have two types of T cells. We have helpers that exactly do what that sounds like, helping, and they're almost like the orchestrators of the immune response. And then we have killers, which again do what they sound like. They go off and find threats and kill infected cells, whether it's an infection, or we also hope that we have immune responses that kill off foreign things like the beginning of cancer, or when cancer evolves and our immune system can find those cancer cells and kill them. So if we go back to what my question was in my PhD, it was the T cells. So I was looking at helpers and killers, but again, the naive ones that have never seen anything. Because what we hoped to do long term is to train the naive ones to say, hey, this foreign protein is part of cancer. And so therefore, can we use that knowledge to train naive T cells to grow into an army for therapies
of cancer-fighting cells.
Yeah.
So you effectively prime them. And so is the end goal with that technique to be able to create the naive cells out of the body, train them, and then inject them?
Exactly.
Wow.
That's called essentially, T cell therapy.
Oh, wow.
So yeah, that's the long game, and how we want to understand more and more about the immune system, the immune response, and how we can train cells. So obviously, as that sounds, we need the naive cells in the laboratory. We need to train it to detect and grow up to be specific for cancer or a virus or so on and so forth. And so my initial role was in my PhD was to understand what are the types of signals that those cells might need in the laboratory to see the signal grow. First of all, we had to find the needle in the haystack, a type of cell that can mimic or, find cancer or a virus or whatever. But essentially my first question was just like, if we stimulate a naive T cell and grow it, what are all the signals it needs? So a lot of that was just drawing on what we know about the immune system and the types of proteins the immune system needs. But that was sort of the crux of what I was doing. So making them grow into big armies and then keeping them alive thereafter. Because what happens in an immune response is, yes, we create armies of T cells, but it's also natural at the end of immune response, when the job is done, you've kicked out the foreign thing, whether it's bacteria, whether it's virus, they contract, what we call contract afterwards. But contracting kind of means that, the job is they die off, and we're left with a memory T cell. So these words that we call them are all quite logical. We have memory T cells, we have naive T cells. And so that was where it all sort of started for me, was understanding how we sort of foster that expansion in the laboratory, giving them all the right juices, if you like. And, so just as by way of a protocol so that it can be applied in different settings, yeah.
And so that's what you do. You get those cells, put them into a dish, and you apply proteins, is it, to them that a cancer cell might produce, and that's to help it identify cancer in the future? It's
more like, so an example would be where, say, we get a whole bunch of naive T cells, and you might digest up or, mush up, if you like, cancer type cells to show to the cells so that they can see if we can recognise them. It's far more complicated than that, but that's it stripped back. And so that's to give it its signal. But then it's the immune cells themselves or their environment that they live in the body that might sense that they need a particular growth protein or what we call cytokines, essentially. These are little cytokines is the name we call the immune-specific molecules that then tell the cell time to grow, expand up.
So they're regulating your immune response.
Yeah.
And that's why you can have a cytokine storm where they go, that's out of whack, is it? And they're telling the body to produce too much fluid or something, and so you have inflammation.
And so when we speak of, it being the intricate sort of immune response, and again, the helpers and the killers, when we flick back to the helper T cells, I almost call them like the orchestrator. And if your orchestra is not all tuned finely, it sounds horrific, right? Same thing with the immune response. So the helper T cell helps tune the killer T cells to do their job at the right time in the right way, as it trains up B cells, which we can also talk about. But the B cells are the cell that creates antibodies. Again, it's quite easy to talk about in the context of COVID, but essentially this helper is orchestrating. So what we sort of understand about, things like cytokine storms or things like that, not every immune response, is favourable. So, that's where something might be done, your body might kick off irresponsibly or, completely in an untuned way, and that's where our immune system can cause harm, if you like. So again, early in the COVID response, the race at the beginning was, we need to understand the immune response to this virus. Do we generate effective memory so that we can fight it off again? Why are people dying? Is it anything to do with an immune response going wrong? And all those are all the sorts of questions that we were asking early on, and when there's a new threat that's emerged. Those are all the sorts of questions that we dive into.
Yeah. And did you get some answers to some of those questions? Like, what was the issue with the COVID virus? What was it doing in terms of the immune system?
Yeah, so and I say the global we, not us specifically, the international researchers that were going hard in the space, were that we knew that as we age, that people as you start to age, your immune system starts to not be as great. And that's like with everything when we age, right? So, and that's because we sort of are born with, as I sort of described, naive cells, and we spend our whole lives fighting off threats. So one of those questions was, is it because we don't have any cells left that can fight off a new thing? Oh, yeah. Or is it just general, falling to pieces of our bodies because that's what happens with aging?
The system's wearing down with age.
Yeah, exactly. Yeah. And one of the core papers that came out in 2020 discussed this, as I just described, the orchestration of the immune response, that it was just not fighting in the right sort of pattern, or so
the helper T cells weren't working as effectively as when you were younger, perhaps.
It kind of thing, like as in they called it almost unorchestrated. Like you might find that yes, you've got killer T cells and yes, you've got helpers, but it all was kind of not in sync or working the way it should have, and therefore that's where some people could succumb. I mean, the virus itself, especially pre-Omicron, was much more targeting the lungs, which was very catastrophic, which is why the rest is history, right? But essentially that virus back then was targeting the lungs. And so all those early questions were, is it that the virus is causing damage, which is leading to mortality? Or is the immune system overreacting, which is causing mortality? And some of those things are not necessarily certain right now, like as in when we don't necessarily say that this person had an overreactive immune response and this one didn't quite make it, kind of thing. But from there, that's where, treatments emerged as well. So, the big race for antivirals to stop the virus doing damage, obviously vaccination to prime our immune system first so that it's ready to fight faster.
And that's to try and get the T cells to go out and then turn some of them into memory cells after the response has gone down and generate those long-term memory cells, I guess?
Exactly. So when we sort of think about the job of vaccination, whichever the threat is that it's training the immune system in a safe way. So if we look at, again, the SARS-CoV-2 virus, there's something like 29 proteins, I think it is, whereas vaccination is using just the one spike protein. So what happens is our immune system sees this one spike protein and gets trained, and it's like, okay, next time I see this is a threat. Yeah. So with immune memory, and why we need multiple doses of vaccines as well, is that the first time it's like, oh, this is new, creates an immune response. They grow, they die off, and you're left behind with memory T cells and memory B cells. The memory B cells are actually really critical in stopping things like viruses because they make the antibodies. The antibodies stick to the spike protein, which is what we call neutralizing antibodies. Yeah. So when you inhale the virus, your immune system kicks in really quickly. A core aspect of immune memory is quicker, faster, harder. So with vaccination, you get primed and you see that first response, but it's really the second time you see it that your body's like, oh, yeah, I really need to make a solid, good army because, against this threat. And so that's what our 2 doses are. Obviously, the complicated factor that we then faced was, in a pandemic virus, it's constantly evolving and escaping our immune responses.
Yeah.
That's literally what's going on there. So, that's why, that's why we're still where we are, because the virus keeps evading, and we hope that at least our core memory response from
sticks around, and so
exactly.
And if it's close enough, if the change isn't too much in a variant, then your body can detect it and still deal with it in general.
Exactly.
And I guess too, your body's immune system will be able to, once it's had an infection, it'll break down all 29 proteins and probably get a better overall look. It's like, okay, I can see 29 proteins, so if 27 of them match next time, I'm pretty sure this is a foreign object, so I will produce the response to it.
Yeah, exactly. And that's sort of the term that we hear a bit at the moment is hybrid immunity. So the thing that happened here in New Zealand is that we had really high vaccination rates before we had a really, our first proper wave of COVID-19 infections everywhere, right? So we had, when Omicron hit early 2022, we were highly vaccinated. And so what that sort of showed us, and it continues to sort of demonstrate that, is that your immune system fought hard and fast quickly when it did see the virus. And then because your immune system's breaking down that virus, and as you say, it's seeing all the proteins of the virus, it's training it to be, to recognise a whole virus instead of just the spike protein.
It's amazing the way that the cells do that. I can't think of the name, if they're macrophage or what they are.
Yes,
Is it macrophage?
Yeah.
That take a cell, take it in, break it into parts, expose parts of it onto the outside of the cell so that other immune cells can come along and go, okay, you're telling me this is a foreign thing. If I see this again, we know. It's just insanely complex.
Exactly. That's exactly what happens. So, our specialised immune cells gobble up foreign objects and pop the proteins out on rubbish bins on the outside. And our cells are doing that all the time.
Yeah.
We're always breaking down proteins and popping them in rubbish bins. And our little patrollers, our T cells, our B cells, all our immune second defenders, if you like, are constantly patrolling. And so when it bumps into a rubbish bin with a foreign object in it, it says, oh, this is foreign, let's kick in. Yeah. And so, as I say, that can happen with cancer cells too, or it can happen, so basically anything foreign. The tricky thing about cancer
is that it's not foreign.
Exactly. Yeah. It's not foreign enough. Yeah. Exactly.
So you need to find some other thing about this cell for the body to recognise that, yes, it's part of you, and it looks like maybe it is a blood cell, but this thing that it's doing differently you need to pick up on. Exactly.
So, the viruses are obviously foreign, right? So, but yes, with cancer cells, it's where a healthy cell's gone rogue, right? So it can produce other proteins that it's not supposed to, but it's not foreign enough. And so what cancer immunology is often about, or, it's focusing on harnessing the immune system to fight cancer, is about trying to, screen cancers to find, proteins that are specific to the cancer.
Yeah.
And it might be that it's a human protein, but basically we're trying to find something slightly foreign because the T cell response is so clever, so intricate and so specialised that it'll only kill off, either infected cells or cells that are foreign to us. And so it's incredible how our immune system works. But that's the hard part with cancer.
Cellular biology is just amazing. The more I look at it and read it, just the more amazed I am about how each of those cells is a very complex machine with many very complex little machines inside of it.
Yeah.
And just operating with the most amazing engineering because they're not like we see, nuts and bolts and bits of steel off here and electrical power running through it. No, these things work purely by the shape they're fashioned in and the way that electrons attract one another, perfectly formed, and they do these jobs. Like, what's the thing that reproduces mRNA? I can't think of the name of it. It's a little unit in the body, and it's apparently working at something like 10,000 RPM, spinning around, taking in bits of food or nutrients from the cell, producing RNA out the other side or DNA, whatever it does. It's just amazing.
Yeah, our cells are incredible. Yeah. Absolutely, and it's one of those things. Like with the immune system, it's so complicated, right? But our immune system often fails us too. There's lots of illnesses that we can get through I say failure it's gone wrong. Autoimmunity is when
Yeah.
our immune system starts attacking ourselves.
Yeah.
Allergies are such a pain. I mean, I've got a long personal history, and how annoying allergies are as well.
So,
all of these things.
So is an allergy, though, is that your body actually working? It's like, hey, you've got pollen in your face. We're just going to try and flush it out, but we suffer it because you've got inflammation. Is that actually working? Like an allergy, is our immune system working there?
Kind of like it's where and there's sensitivity. It's quite complicated, right? Like there's sensitivities to particular antigens, whether it's food or whatever the thing we're reacting to. There's sensitivities, and then there's full-blown allergies. If someone's hyper allergic to something, you go into anaphylaxis, and it can kill you. And so again, it's where your body has seen that protein at some point, and it's developing
A really strong response. Aggressive is a good word.
And a response when it's kind of like, hey, don't worry about that. It's only pollen. You don't need to fight back. But once the immune system's trained to do that, it doesn't forget.
Yeah.
So those are the sorts of things that make our immune responses so complicated.
As you were saying before with cancer, the tricky thing about cancer, when I think about it, all of the strategies to treat cancer that I can kind of think of, radio and chemo and all those kind of things, they're broad. Like they wipe out all the cells to get the cancer cells, because you can't just target those cancer cells.
Yeah, often you hear the term that we don't have a cure for cancer. We cut it out. Yeah. So we cut tumours out, and then exactly. A lot of the strategies we have are non-specific. That's why you get lots of side effects from some of these things. So if we can train the immune system to be specific. And it's obviously not straightforward, is it? Otherwise, this would be everywhere.
Easy.
But it is happening. Like there are, there are therapies that are evolving, and we'll hear more and more about this, moving forward.
And that was, as you mentioned before, that's a T cell strategy treatment.
Yeah, because that little receptor on the T cell, that's on the receptors on the surface of T cells is so specific, like it recognises things, in an intricate way. So if we can train them to recognise something foreign, then that's one of the strategies. And the key thing is having those specific proteins that are expressed by a tumour or cancer cell. And that's the tricky part.
And there are some things, though, you've mentioned proteins before. Do some cancer cells give you that hint that they do something a little bit different to what they should be doing?
Yeah, so, and the tricky thing is to make sure it's only ever a cancer cell that's expressing it. Yeah. Because you don't want these little army of
then you've got
killers.
an autoimmune issue going on again.
Yeah, well, if you target the wrong protein, they'll start attacking you. A perfect example, actually, is with melanoma. So early studies in this sort of space were amazing, so melanoma is, a melanocyte gone rogue, essentially. So, the melanin in the skin so the melanocytes turn cancerous, but the melanin in the skin we can target with a T cell. And it's one of the tools we use in immunology because we can find them quite easily, I'll say, compared to others. Like, as I said, a naive T cell is like a needle in a haystack. It could be 1 in a million, and it's really hard to find them. So one of our model antigens, is what we call it, can target melanocytes, or the melanin in the skin. And these were the first sort of trials in this space, because you can train a T cell to go and fight melanin specific. In melanoma, basically fight melanoma. But the problem there is, if they're good, it'll wipe out any cell that's got melatonin in it. Our eyes, so essentially, when the first sort of papers started coming through it, how amazing T cells were at being specific. Yes, there was a great examples of serious tumours getting annihilated by these T cells, and and you're now alive. And in fact, the eyes are protectable. I think I'm just racking my brains here because it was a while ago since I remember reading this, but I think you can use a steroidal type response in the eyes to sort of say, hey, leave my eyes alone. Okay. But still, it was it wasn't specific enough. The response was specific, so it's knocking out your melanocytes, but it sort of demonstrated how amazing, you know, that T cell response is. Yeah. And so like some of those, as I say, those early publications were showing, you know, you just completely wipe out all your skin colour.
Amazing tumour results, but there's some side effects.
Yeah, exactly. And so it was from those early studies that it's like, look how amazing the T cell response is when we can train it. But obviously, we want to train it to fight in a way that's not going to damage, you know, have off-target effects. So so that's just sort of one example in that space. Yeah.
So there's different types of cells. The T cells, the killer ones, do they kill the cell like a macrophage? Like, do they go and attack it, break the walls down, or do they tell it to kill itself? Do they signal to it, tap it on the shoulder and say, hey, it's time for you to stop?
Yeah, what they do is they snuggle up because the rubbish bins on the outside, you know, they recognise this protein, and the receptor on the T cell recognises that and says, you need to be gone with. And what it does is...
So just to step back, when you say the rubbish bins on the outside, we're talking, there's a cell. The cell has expressed what would normally be considered rubbish onto the outside of the cell. T cell goes past and says, normal rubbish, don't worry about it. But now it sees something foreign and it's like, okay, there's something wrong with this cell.
Like viral proteins, right? So, you know, when a virus infects a cell, it's going to chuck out the proteins just like it does with good proteins. And the T cell says, oh, that's not us, that's foreign. So it says, right, you must die. I must get rid of you. And so it pokes holes in the cell and shoots in toxins.
Does it?
Yeah.
Wow.
With intricate, specificity.
Yeah, that's insane.
Yeah, it's so cool. It's so cool. And we can see this, you know, in the laboratory as well. Like, you know, we've got cool videos where we have a cell line that's specific for cancer or something like that. And you throw in a T cell that you've trained up to kill that particular cell, and it snuggles up to it, sniffs it out, snuggles up. And when you sort of see a video of them, they're like little worms creeping around a cell going, oh, you're foreign. And then it pokes holes in it and kills it. And then they apoptose and, you know, they all starts to bleb up and become a big dying thing. And then the macrophages will come along and gobble up the rubbish and so on and so forth. So that can be how you keep that response going.
Amazing. I think it's the video of macrophage that I saw the other day chasing a bacterium.
Yeah,
it was
so cool.
It was like this cell was just chasing this thing around. It's mind-blowing. How does it do that? Yeah. It doesn't have eyes, doesn't have ears. I don't know if it can feel. How is it tracking that thing?
It's amazing, isn't it? When you see videos, they literally look like they're alive creatures. Yeah. It's so incredible. Yeah. Like, you know, sniffing around going, oh, no, I'm going to chase you down. And it's often because it's often called what we call chemotaxis. It's going towards a signal. Yeah. Okay. So, you know, and those are other big questions in immunology. You know, does something move towards a signal, or is it random and it just bumps into things? Those are literal things that, you know, we study.
And then again, if it is chemotaxis, if it is moving towards a chemical signal, again, so that's effectively that system is being organised by the laws of chemistry in that, you know, the covalent bonds on this particle and this particle here will attract one another, and that's going to effectively pull this thing towards it.
It's so cool.
This is what 4 billion years of evolution does.
Yeah, it's so cool.
Because, I'm sure there are inefficiencies in areas in our cells, in our bodies. Like you can look at something and go, well, that's an inefficient process. But I bet you overall our cells are extremely efficient purely because so many different processes happen within them with the machinery that's in there. Yeah,
absolutely. And that's why, you know, like one of the things, you know, when I sort of talk about our immune systems failing us, it could be because some of the building blocks aren't there, right? Like just like any organised system, it will go slowly or not, or it'll sort of fall apart. If it doesn't have all the building blocks, and you know that's our vitamins,
nutrition,
our nutrition. Exactly.
Yeah, exactly.
It all starts with nutrition, and that's why that side of things and our health is so important about driving good immune responses. It's not to say that it's our fault when it goes wrong, but you know, of course, you know,
I think there's the French have a concept of this in health, which they talk about like preparing the land, where they're talking about exactly that. It's like if you want to be able to fight infections, well, prepare the land, like prepare your body well, have the right vitamins that you need, have general good health so that your systems are capable of functioning well, which I guess in terms of viral response means you've got the energy and the sleep and the nutrients for that machinery to spin up quickly and orchestrate well and operate well.
Exactly. And that's my personal approach to, you know, keeping healthy and keeping these viruses at bay and everything is making sure, it starts with nutrition, it starts with gut health, all of those things. And it can be just tricky knowing exactly what helps what. And, the microbiome that lives in, we've got microbiomes everywhere, but essentially gut health is so huge. And it's also amazing how embarrassingly recent
that knowledge is.
Oh, and we're learning all the time, right? But it's crazy that it's taken this long to go, our microbiome. So we're full of bugs, right? Like I often say, we're an apartment block wandering around, getting controlled by all these species, and we actually have to we're the landlord, right? We have to keep them happy. And sometimes we get bad neighbours, or, you could have a whole apartment block that is full of, unsavoury
get rid
characters.
of those tenants and get some new ones in.
And it's so difficult to try and sort of foster the exact species we need or neighbours we need. And we're still actually learning. As I said, it's embarrassingly slow about, how much we know about the microbiome. But I remember being at an immunology conference and something came up and I'm like, this is crazy. We've sort of known this in passing, but here's the scientific evidence. And so our immune systems are so reliant on our gut microbiome and, and our even mental health. But, there's so much signalling that goes on in our guts.
Yeah.
So, there's so much we don't know.
Oh, and the amount of information that goes back from the gut to the brain. And personal experience, I changed my diet pretty severely a couple of years ago where I went and just did the carnivore diet. So I went from eating everything and eating a lot of shit, a lot of bread, a lot of sugar and all that sort of thing to eating nothing but meat. And within 4 days, I noticed a dramatic drop in anxiety is what I noticed. And that stayed like that. As long as I was on that diet, I just didn't get that anxiety anymore. And I firmly believe that had to do with changing the gut flora. That had a big part to play in it, I think. The other thing that's really interesting is around like introducing the right bacterial agents in your body is for people who have got gut issues and they do the fecal tablets where they take someone with a healthy biome, take a tiny bit of feces, bring that into their gut and let it flourish, and now they've got the right bacteria growing again rather than something they were deficient in. Which again makes sense.
I think I think we're going to see more of that.
Yeah.
Like, I think we're just taking a long time to catch up.
Yeah.
But like exactly what you described, like I've been down that personal path as well. I've had so many problems with allergies and I was sick of it. And I didn't want to just, take the approach of masking symptoms with antihistamines and everything because there wasn't anything specific that I was allergic to. And, so I did all the patch tests and everything, but it didn't ever answer my question. And so I learned so much just from a personal perspective about the foods that you can and can't eat. And so I started gluten-free, and that because I had this horrific skin condition, and when I went gluten-free, it cleared it up. So that was kind of like where I started, and it was such a long time ago. And I was just like, it was that realization of how powerful what you eat is. Yeah. But I think one of the things with humans is we're impatient, and we're used to traditional medicine and pills and drugs and quick fixes. Yeah. So, and I often talk about this, when, like these are completely, N=1 things that I do, or, that we often chat about, things that help. But diet, I think, has been such a huge change for me personally. Yeah. And as I said, there was no real reason why I had this manifestation of cracked, dry hands and everything, but it cleared up instantly when I cut gluten. And then I just sort of continued. And I also noticed massive differences when I went from what I thought was low carb and then sort of even, getting rid of all the crap, as you said, the no sugar and everything like that. And it's been a game changer for me. Yeah. Like, so now all of my own sort of stimulatory or allergy type symptoms are so manageable now. Yeah. So manageable because it was all about what triggered me. And it wasn't because I was allergic to that thing. Sometimes it's just a bath filling of being stimulated by the wrong things. Yeah. And for me, it's not that every human's this way. And that's the other thing. It's always about what works for you or the journey that you go on to, feel better. And so that's why, as I say, it's not like a one size fits all. It's kind of like saying what you need to do to reshape your microbiome to get on that path that feels good. Yeah. And that's essentially where I've got to.
I think even one way of looking at it is simply it's a numbers game. If you compare eating whole foods, let's say, like meat, eggs, fish, vegetables, fruit, nicely grown well, to processed foods, just the processed foods which contain all those whole foods and a whole heap of additives, you're just reducing all those additives out of your diet. So all of those things, now that if a lot of those additives probably aren't seen as natural items to your body and perhaps aren't the things that we have evolved with over the last at least couple of 100,000 years, fruits and things and meat and all that to eat. So your body naturally says, well, I've got to start inflaming the gut a bit because you're putting all this stuff in I'm not used to. Yeah. So you've got constant inflammation going up, travelling through the blood-brain barrier, making your head foggy, all this sort of stuff. Diet's huge.
Diet's huge. And I think we're just in the early stages of it, I think. But it will always be tricky because it's not a quick fix. You know, how long does how long does it really take to feel enough of a change and go, yep, this is my new way.
And to stick to it.
Exactly.
And if it's sugar you're trying to beat, I was talking to a friend the other day on the show, sugar is the toughest drug that I've ever tried to stop.
Yeah.
Like no sugar, like actually stop it. You have no sugar in your diet, man. After 2 days, I'm just like, I'm just dreaming about a block of chocolate or, fuck, I could eat a whole bucket of ice cream. I could just do it now. Because it's the toughest drug. It's only been around for 300 years. Well, at least Western world. I don't know, in developing countries before that, if they were using it a lot. I think it was about the 1700s it turned up in Europe and was brought over here. So we've had 200 years to get used to sugar and it is insane in the body.
Yeah.
It's tough to stop.
I've kicked it.
Have you?
Yeah.
Oh, good.
I'm nailing it.
I'm not. I've got it ring-fenced to blocks of chocolate each week, but that's about it.
I eat 95%
dark chocolate.
Yeah.
You're a psycho. I can't do that. That's just too much. My wife's the same. She's like, dark chocolates. Oh, no, give me those sugary ones. Can we give the caramello?
My poison that still works. Like I do have the odd sort of sweet thing that's with all the, the sweetness that everyone hates. But because it's been such a long time since I've had proper sugar, I don't really notice. So, and they're just as bad for you, but like that's my crutch. Like if I like some marshmallow made with fake sugar and I'm like, I need to pop one of them. Or because 95% dark chocolate is still, is fine for me.
Yeah, well, that's the thing.
So it's good.
It's got all the chocolate benefits. It just doesn't have the sugar. That's it. Yeah, I think that's it. People who eat dark chocolate don't really have that sweet tooth, or they've kicked it because you're eating it for a different reason. You're eating it for the bromines or whatever it is in the cocoa.
And I feel it, like I'm like, woo, this chocolate's great. And so absolutely. So all of those processed foods, is going to, some of us can't handle it. And that's what I say. It is a personalised approach to, because some people can eat anything they like and they're fit as a fiddle and that's great. But I think when you recognise that you're someone that's reacting funny to things or you've now responded to things, that's where it can really, really help. And that was my journey. And so, like even within my family, they sort of go, oh God, here comes the fussy one. And it's not been about being fussy. It's just my journey that works for me kind of thing. Yeah. And so, one of the things with our immune system and why, the sort of irrational responses or irresponsible or wrong responses our immune systems kick off. Again, it comes back to viruses, or one of the paths can be from a viral infection. So, things like autoimmune diseases can actually, it's not really proven because nothing like this can be solidly proven. But one of the ways that we understand autoimmunity is that your body's fighting off a virus, right? And we're doing it all the time. We see viruses from a young age. Little kids, right? They're always sick, sneezing and coughing. We're constantly being exposed to viruses. And one of the rationales or potential understanding behind autoimmunity is that while we're fighting a virus, we accidentally see proteins that are self or food. Yeah. And we start to create immune responses against those things irresponsibly.
When we shouldn't
We should just ignore those things. So that's one of the rationales behind, You hear a bit about people who, even with celiac disease, right? Celiac is, responding to gluten peptide, right? So it's literally like, oh my goodness, here's a foreign object, and you start to attack self. Like, that is literally what's going on there, right? And some people don't know that they have this that can be like in later life. And it could be because, your immune system hasn't been in that situation where it's seen gluten in a strong enough way to create a full-blown response. And then it could be, that could be what triggers it later on. But I mean, once you're celiac. It's not to say, that you can switch that on or off or anything like that. But essentially that could be when the actual illness is triggered. Could be, as I say, it's kind of a hypothetical situation.
Yeah.
Could be while you're fighting off an infection.
An infection, a virus of some sort. Yeah.
Yeah. So there's a couple of, autoimmune disorders that are generally sort of linked to potentially kicking off, when you're fighting off infections or infections in general. Yeah.
Have you done any work, or have you seen any work on resetting immune response, so that if you have, you've detected you've got an autoimmune condition going on, like your body's attacking your thyroid or whatever it shouldn't be, can you sort of is there a way to tell the body to go, hang on, this is actually you, stop doing that?
We would get rid of autoimmunity if that was the case.
Well, yeah, I guess so. But I was, because I know I've read people who've tried to like do fasting, because apparently if you fast for a period of time, that does something. It cleans up a lot of the autoimmune responses. It reduces the immune response to some extent, which I guess is because you're actually not triggering it. You're not eating anything for 3 or 4 days. And so your body's like, well, we don't need to be, there's nothing here triggering us. And then maybe that gives the ability to somehow reset some.
So some of that could be, say, I'm just trying to think of a particular example, but with some of our autoimmune conditions that that are linked to being triggered by diet, or something like, I'm just trying to think of an example and I'm struggling, but essentially, let's say some of these proteins can be quite stimulatory. Like, you often hear people with autoimmune conditions feeling better with going gluten-free or wheat-free or dairy-free, all of those things. And it might not necessarily be that you're allergic to those things. So, but it could be that you do have,
reduce the amount of stimulation anyhow because of taking something out.
And what can happen, as I say, I can't think of an actual example, but what can happen is another phenomenon called cross-reactivity. And it's where a T cell that normally fights off, or, has normally fought off a virus, the proteins from that virus are slightly similar to, let's say, a food product, let's say one of those typical ones. And therefore you've got an immune response against, those things. Yeah. Again, it's not necessarily an allergy. It's more sort of referred to as sensitivities, where you just might react when you eat those things. Yeah. So, because obviously there are full-blown allergies to these things, full-blown. But sometimes those sensitivities can be, and again, hand wavy in a hypothetical way, be linked to a food that you might now be sensitive to. So when you fast and all those sorts of things, your body's not seeing any of those antigens, and it can be quite calming. There's loads of things that go on with fasting. I'm a big fasting fan as well. So yes, there's all those sorts of things that go on, and that can be considered like a reset, right? Because your body has not been seeing any allergens or
been activated
proteins. Yeah. So that can be what sort of links to a so-called reset in that regard. But yeah, as I say, there's lots of other mechanisms that go on when you fast.
So in some of the other work that you do with, like around chronic fatigue and long COVID, those or maybe not long COVID in this question, but chronic fatigue, then is one of the reasons that the body has some constant inflammation going on? Because I imagine, again, just from a engineering mechanical perspective, if your body's seeing inflammation, it's got to be burning some level of energy to be dealing with that, and it's triggering your immune system. If it's doing that all the time and that's sending signals to your body, that's a whole heap of workload that you just don't need to begin with. So if you were to, if your diet was to change, you take that away, then suddenly, I'm just wondering if you start to get some energy back anyhow, because your body's not effectively thinking it's constantly fighting something off.
Yeah, so let's talk about those things. So those illnesses, so myalgic encephalomyelitis, chronic fatigue syndrome, ME/CFS. It's such a mouthful. So ME/CFS has been with us for decades and completely understudied, neglected both in research and medically. So here we are with a new virus that's triggering the same illness. And so there's a massive overlap with ME/CFS and long COVID. So if we strip this all back, there's a few things to unpack here, but essentially, well, let's start with that fatigue situation. So fatigue, we hate the word fatigue with these illnesses because it sounds like tiredness. And even chronic fatigue itself can be quite different to the syndrome. So, because chronic fatigue can be aligned with many states of illness, because it's common to be in a fatigued state with particular illnesses outside of this particular actual syndrome. So what people describe with the fatigue state, they almost sort of will tell you that it's like being poisoned. And it's so difficult to articulate what this really, really feels like when your body cannot produce energy.
Like being hungover, I imagine.
Potentially, but even, in fact, I think the hangover feeling people have actually described with this illness, more relating to the brain fog feeling, but the fatigue is so deep-seated and so indicative of a major breakdown of some pathway going on in your body. So that's why we need to sink our teeth into the research. And yes, there's a massive overlap between long COVID and ME/CFS, which is the area of research we're focusing on. So our big question is, what are the biological mechanisms going on? What causes these illnesses? So obviously when a pandemic virus hits and you start to see a wave of people saying, hey, I'm not recovered, and it took a lot of loud voices. And I often talk about this, that in 2020 when, internationally when the waves hit, sadly, it took medical doctors and or athletes to raise the alarm to be taken seriously. And so because it was like these illnesses have been disregarded in the past as not really a biologically triggered illness. I
guess that's why, because in some respects it's easy for people to dismiss them, to say, well, where's the proof that you say, well, what's going on? Yeah. And if it's something that you don't understand, it's like, well, I can't tell you, man, I feel like shit all the time.
Yeah, exactly. So there's no diagnostic tests for these illnesses. There's no there's tons of research in long COVID now. But essentially there's no diagnostic tests and there are no treatments and there are no cures. All of those sort of things are what we hear about all the time. So when SARS-CoV-2 was causing this, and as I say, it took these, groups of people being impacted to sort of yell from the rooftops. And people with ME were like, yeah, welcome. Yeah, hi.
Is this your first rodeo? We've
been here for decades. Yeah. Now people are listening. So essentially what happened there, and the reason why there were so many healthcare workers, I mean, it's not rocket science, right? Healthcare workers were not protected.
We're being exposed.
They were getting exposed left, right and centre. Yeah. And they were not really getting, they didn't have the full-blown protective elements we know to keep a airborne virus at bay. Yeah. So especially, in the UK, around the world where the waves really kicked off, it was tons and tons of healthcare workers who were impacted, right? So, and I know many of these, that are still suffering today. So what this told us, if those of us that had never heard of these illnesses, is that a viral infection can trigger a chronic illness, right? And so historically these illnesses were brushed off as being psychological, because if a test we have in our clinics can't pick up your disease, it must be in your brain.
We'll ship over there.
Yeah, which is just so dire and so frustratingly annoying, right? Like, just because it's not in your medical textbook, do not disregard this. So we call that the silver lining of the pandemic, that it's not great that there's tons of people suffering, but the silver lining is bringing it to the attention of people who have dismissed this.
Put some focus on it.
Yeah. So what this has told us is a number of things. It tells us that viral infections can switch on pathways that then don't turn off or, trigger an illness. And then it's bringing back into the spotlight. Let's start, seeing what ME/CFS is as well. And I mean, the massive overlap there is that viral infections, not just viruses, but infections are generally one of the most known causes of these illnesses. Obviously with SARS-CoV-2, yes, it's causing long COVID.
So people with chronic fatigue syndrome have often indicate that, hey, I've had glandular fever or I've had something, and then this doesn't seem to heal, even though I don't seem to be sick with it anymore.
Yeah, so that's where we're headed. So I think the statistic is something like 80% is linked to an infection. And I would argue they probably all are, and you'll sort of see why I think this. So interestingly, many people so we're doing a long COVID study, and we're also going to overlap with ME/CFS, and many people that would turn up, to donate their blood to be on my study, very, very sick. And, they chat away because they're just like, this is just incredibly debilitating. I can't believe I feel this way. And, have a chat about what they're going through. And coincidentally, the number of people when discussing with me how horrible they felt, they were like, oh, this is, the last time I felt this horrible was when I had glandular fever. And I'm going, like, and it's not across the board. There's plenty of people who had glandular fever who haven't been impacted by SARS-CoV-2, but there's a strong correlation with people with long COVID sort of symptoms where they're not recovering volunteer that they were the kid that got glandular fever.
I remember at school that glandular fever was one of those things where it was like, hey, you don't really want to get that because there's a good chance you'll be out for a couple of months if you get glandular fever. Yes,
so Epstein-Barr virus, EBV, is the culprit that causes glandular fever. It also gets referred to as kissing disease because it's when teenagers start kissing each other that, and it's that age group that spreads it. So what the statistics tell us is that something like, hand wavy again, because when was the last time anyone checked, but they sort of say that 90% of us have been exposed to EBV, right? So what the next layer on that is that only some of us, I can't remember what the percent is, but it's not a high percent, get glandular fever. What that's saying is that the rest of us get asymptomatic infection. Yeah. So that's why when we're all sharing saliva, and I mean, you can share saliva through, households with children, a lot of kids will get exposed at a much younger age anyway, just by sharing cups in the home and all the rest of it. But it gets that cute little phrase, kissing disease, because that's when it really spreads around.
You tend to see it, yeah.
So it's in our saliva. Yeah. And, that's how it spreads. So that tells us a number of things. That virus we all get, pretty much. Only some people get symptomatic infection and it kicks their butt, glandular fever, and it's strongly correlated to ME/CFS. On top of that, that virus is known to cause cancers. And again, only in a small percent, because any virus that we all get and it causes a cancer in only some, it gives us hints and tips that causes cancer.
Just on that point, so that virus is doing something to cells to tell those cells to then trigger them to go and mutate, like changing some process so that they're mutating and becoming cancerous.
Yeah, I just missed a bridge there. So when we get infected, we try and shut it down and kill off the infection. There are some viruses that stay in us forever, and that is one of them.
Right.
So that's the key point here, is what we call latent viruses or dormant.
Yep.
So the big elephant in the room of all of these illnesses is that I think we take for granted that our immune systems on a daily basis when we're wandering around healthily is keeping all our latent viruses that live on board forever
under control.
It's incredible that we do this. So it's the herpes family are the culprits. We get infected and they're with us for life. So the classic ones we know about is EBV, is a herpes, it's called human herpes virus 4, but we know it as EBV. Then we've got another one called CMV or cytomegalovirus. I hadn't even heard of this until I got my butt kicked with it during my PhD. Like when it was a virus that we studied in the lab, or, was part of our experience. And I'm like,
oh, hands-on experience.
I was like, what the hell is this virus? And oh my God, it kicked my butt. And so I got my butt kicked in a glandular fever type way by that one. So cytomegalovirus, CMV, never heard of it. And then the other classics are your herpes simplex 1 and 2, cold sores.
Yeah.
And then chickenpox.
Yep.
Chickenpox that then stays on board and comes out as shingles. So those are the family of culprits that we take for granted that we keep under control.
And which is why I guess, and I'm just guessing here anecdotally, that the reason that sometimes shingles turn up in adults is because you've worn yourself down a bit. Your immune system perhaps is not capable of keeping that virus under control, so it flares up.
So that's exactly right. So when we think of our latent viruses, and again with these illnesses, so the pandemic's shown us this too. Viruses that present on our skin, so shingles, cold sores, that's displaying that you've just had a reactivation, right? Again, it's relevant to this pandemic because, I will be standing there giving a presentation saying this, and there's lots of nods going on in the audience, whether it's healthcare workers or patients and things like that, and especially healthcare workers who are like, we're saying tons of shingles now, or now we're saying this, and this. So you get lots of nods going on. So again, coming back to ME, and, someone will say, I know my glandular fever was my trigger point for my illness, or so on and so forth. But then it might be something like a stressor event where they think that was related to their onset of illness. And now my thinking is that, sure, you might have a stressor event, like as you just described, whether it's, kicking your body where you're not sleeping enough, poor nutrition, those stressor events. Even stress is not just a term and a feeling. There's lots of molecules getting released when we're stressed, right? So once our body is so-called stressed, again, it's impacting our immune system, and then you get a reactivation potentially. And so, as I say, with this pandemic, people described getting shingles after their infection. When I was chatting to colleagues at work, and they're like, I had cold sore outbreaks for so long afterwards. And this is interesting that now you're saying that it's probably linked. So
something suppressing the immune system there.
Well, what my sort of hypothesis with all this is that SARS-CoV-2 is a horrible virus that kicks us in such a way that we're in a state of unknown length of a little bit, I don't call it immune suppression, but we've had a whack to our immune system, and it's going to take us a while to get back to what I'd describe as a baseline.
Yep.
And so during that trajectory of an unknown length, probably very different in everyone, we're sort of vulnerable, and we might get viral reactivations, and we'll see some of them. We'll see our shingles, we'll see our cold sores. And the other ones we don't present, so if you have an EBV reactivation, we don't know what that looks like. You might be, as in the true sense of another way we sort of describe it as a smouldering reactivation. And the reason we say why this is so challenging is we don't have tests available to say, yep, your virus is reactivated.
Yeah, right.
And so full-blown reactivations that can lead to illness, say in a truly immunosuppressed person, whether they're getting cancer treatment, bone marrow transplants, those are other sort of examples of when our immune systems are completely suppressed, and those what I call boring viruses that we live with can actually result in killing the patient.
Yeah, because there's nothing to stop them.
Because there's nothing there keeping them under control. So what's really, really difficult scientifically is trying to understand and unpack and therefore detect when there's sort of a smouldering, problematic reinfection. Oh, I keep saying reinfection. Reactivation of these viruses. And so when something, these viruses are controlling our cells, right? So these viruses live on board in a latent, a lot of the term latent kind of means I'm hiding from the immune system. As we've just described, the immune system fights when it can see a threat. Some viruses are clever that they turn off the machinery inside a cell so that they look like they're not there. So that's what...
They're not expressing the proteins to the outside.
So, and that's sort of called latency. And there are some viruses, we barely even have a good understanding of what's actually going on.
Yeah.
And for me, what we're really interested in is what has happened to the immune system, your helpers, the helper T cell that keeps the killer's trained to kill, and to keep the B cells in check and tell the B cells to go and make antibodies when they need. What's happened with that brilliant orchestra, that the orchestra's falling to pieces, or the conductor's not doing their job, and the latent virus comes out, but not in a full-blown, you're really, in a noticeable way, but in such a way that it triggers an illness.
I wonder if it's more like cicadas that like lay dormant for 17 years. And so these viruses, maybe like they've evolved that if we pop up regularly and just all the time, we're just going to get whack-a-mole and eventually they'll get us all and we'll die. But if we have some irregular period of time where we just lay dormant and the body forgets about us, maybe it's been 2 years now, and like the memory cells, perhaps there's less of them, less inclined to remember them, and then it's like, now it's time for us to go again. And so they make another break for it. And so maybe your T cells are working, but like you say, the virus is hiding. They can't see them.
They're hiding. And I think there's some breakdown that's caused the virus to come out. Because why does the virus want to come out? Because it needs to spread again, right?
Yeah.
So that's the whole rationale. The virus ain't thinking, right? It's hijacked us. And so when we get some form of breakdown, it comes out. So why is all this relevant, and how is it relevant to the fatigue state and all the rest of it? Is that the other cool part of our cells are our mitochondria, the little powerhouses, right? So
this was on my list to ask you about.
So the little powerhouses, the little mitochondria, I would put money on that the fatigue state is to do with the mitochondria being a bit broken.
Makes sense.
My money's on it, right? That little powerhouse literally is to keep our energy systems going.
Yeah.
And I bet it's gone wrong.
That would be the first place to look.
Yeah. And the thing is we already know that viruses can cause problems with mitochondria. So it's kind of exactly right. So it's those are where the big questions are. And, we can look through the literature where people have been burrowing away and showing some of these things. But it's going to take a lot of time to go and screaming from that rooftops and really categorically showing, look, this person's had this virus and it's screwed their mitochondria.
Well, it's tough too because, maybe you could get lucky if you did some sort of a study where you said, okay, whole heap of general population, can you please give us your blood? We want to look at a baseline of, and I don't know if you can even do this, but what's your general mitochondrial energy output? And then for people that in that group get CFS or something, can we have a look and can we go, oh yeah, look, your mitochondrial output is decreased, or some process we can actually see
will
has,
be. 100%.
Stopped.
And if we look at some of these viruses for an example, so again, we talked about EBV that causes glandular fever. That infects our B cells. So that's the cells that make our antibodies. Another herpes virus, in fact, it's so understudied, it's still got its boring HHV6 terminology. It hasn't even been given a cute name. So that one, again, I think they say that one's even got higher prevalence in the population. And, we don't even have tests for it because it's kind of like, who cares about that one? I'm saying we need to care about that one. That one infects the helpers, right? And the helper cells are, as I've described, when the helpers, in fact, I've got another example of why helpers are important. Helpers, there's data showing the mitochondria in those helper cells a little bit munted if you do those in vitro, basically by the HHV6 virus. Like it's kind of like all the signs are pointing there. So the example I'll give you and why we sort of think that the immune system, that orchestration of
being dysregulated,
is helpers, as I say, the helpers are at the top here of helping and orchestrating the immune response. So HIV. HIV is a virus that we know a lot about now, in the sense that it infects, categorically, its main target is that helper cell, right? So it infects that helper cell. So, and that's a lifelong illness, but there's great drugs now to keep it all in check. And so what happens, the transition from HIV to AIDS, basically meaning your immune system is now it's falling to pieces, it's now not great. Why? Because those helper cells are dying.
Right.
And so what it tells us, so HIV is a great sort of model of what when proper immune deficiency kicks in, like proper. Like, I mean, I'm not saying this about other viruses, but it's a real demonstration of when that helper cell that has been infected by a virus that's going to start killing it off, what happens when those cells drop below a level? It means that they can't orchestrate the immune system. And you hear, that term opportune infections. So those are the sorts of things that people who are immunocompromised start to get. And then one of these benign illness viruses then kills the person who's immunodeficient, and that's the 8th state, right? So it really tells us how important those helpers are. And of course, I'm not describing that for these other viruses, but all I'm saying is that there could be a dysfunction in the orchestra that's causing, instead of that extreme situation of immune suppression and
death of the B cells,
death of the Bs, so that's the T cell, that one.
Oh, sorry.
That's the T cell. But essentially, the death of the T cell that normally orchestrates is leading to a full-blown state of immunocompromisation. But what we want to try and unpack, and it's going to be really difficult, right, is to say, what does immune dysfunction look like? What's that helper T cell? What's happened to it that it's no longer keeping everything in check?
Yeah.
And is it because in childhood, they've got HHV6, another herpes family, corrupting them? We don't know. And I think the other thing that's going to create, a broad spectrum of different states of illness across the human population is maybe we've all got different onboard viruses or different loads, if you like.
Yeah.
Like for me, CMV kicked my butt, but I know that I had EBV, and I would have had it asymptomatically, because when I was getting tested, because I was so ill, I went through the laundry list of every illness known to man, and they were like, and it was my liver that flagged something was wrong. And it was because CMV inflamed my liver. And they were like, this is weird. You're weird. Why are you so sick? It's summer and what's going on? And so it turned out that, CMV nailed me. But I also knew that at that point I'd had EBV and that wasn't active, and so on and so forth. But we don't have the tools, whether it's clinically or in the research laboratory, to really unpack what's going on. And I think some, and again, it's not going to be across the board, but some of these answers might lie in understanding what our latent viruses are doing.
Yeah. Totally. Those examples are great examples of how some of those viruses are known to be having an impact. I'm sure other ones are quite possibly doing it in other ways. Yeah, and SARS-CoV-2, as I say, it's a nasty virus in that it's not just respiratory, it's causing vascular issues. And this is why, we're sort of being, raising the alarm about, trying to understand more and more about what this virus does to us, because we're early, right? Look how long it was before we understood about the connection HIV and AIDS and all those sorts of things. So we're sort of saying we know stuff already, and we know this virus is nasty in the sense that it can cause blood vessel damage and everything. And most of us kick it out and carry on as per normal. Some of us get long COVID. And on top of that, so just one question. On long COVID, are those people, do they still... Are they in a situation where it's like it's become the virus has become latent, or has it's gone from the body, but they're still seeing effects of maybe damage done or dysregulation? That's where I was headed. So we call long COVID is what we describe as an umbrella term. So we know there's going to be different categories. Right. So some of those categories could be a really, like it might take you 1 or 2 years to recover. And point being is we haven't got tools to understand which category anyone's in yet, but we believe there's going to be categories. One of those categories that's got about a $15 million grant towards it in the US is understanding whether is there a cohort of people that when they're infected, they don't clear it out properly. Yeah. And so no, it's not about latency like the herpes family. These viruses may be what they call in a reservoir. It might be that they're in the gut or, in cell areas. Right. And there's great debate whether the virus, I think it's widely accepted that there's not live virus in people, but there's always going to be outliers. So, there have been studies where, in a very chronically ill person, they may have found live virus, but that's not going to be sort of a mainstream. But what this group are looking at in the US, a massive consortium of people across the US, they're answering the one question, is there viral debris or a viral reservoir somewhere in the body,
which is keeping the immune system active, right? Yeah. So that's the big question they're going after. And I think there'll be a pocket of people. There are a pocket of people that get full-blown tissue damage. So many people who have a severe illness, hospitalised, are likely to have a high chance of suffering for a longer time through the nature of their, how well they handled the primary virus.
And I guess in their case, they might be actually suffering from gross, widespread cell damage.
Yes.
Like they might have really bad infection.
Yes.
Lots of cells died. They're sick from that and damaged from that.
Yes, exactly. So there's tissue damage is how we describe that. There's various organs that can be impacted, and so there's going to be categories. But interestingly, the biggest cohort of people with long COVID had mild illness, and that was what we always wanted to make clear, that you could have hardly touch the sides SARS-CoV-2 infection. Oh, that was easy, and next minute you're not actually recovering, and you kick into a chronic
because it's
illness.
a completely different process. Yes, the infection might have been dealt with well by your body, but
something else has happened.
What it may have done is it's just capable of triggering some change or dysregulation in a process, which without you getting real sick, it happens, and if it doesn't stop, you've still got it.
So I'm in the camp with a couple of other, international long COVID researchers, which is at least for a category, where SARS-CoV-2 is the insult to the body, which unclamps or dysregulates the immune system. You clear it, but something's kicked in that now you have to get to the bottom of. And and we really think that that's often the case for the mild group, like as in, whereas so the sort of the groups, as I say, the groups of cohorts are like severe damage from the virus, where you might not even get the full blown long COVID, but you're definitely going to have a long term illness, like as in a long trajectory, and you might have some of those impacts. Then you've got the classical from mild long COVID, and is that because you've got debris somewhere that your body's not cleared out, or is it because
you have a reservoir somewhere?
Yeah. And so that's what we're expecting. And the reason why that's so important is that anything to do with treatments moving forward, if we lump everyone in together, that's often when drug trials fail, because you've got a different illness. So that's what the race is on, is to understand how can we unpack and sort of categorize.
Get to the real cause, so you can treat the real cause.
Yeah, exactly. So those are the sort of grand hypotheses with, and the relationship between long COVID also and ME, is that, for argument's sake, it could be that all illnesses are caused by a herpes virus, all of them, and that just when your body gets whacked with SARS-CoV-2, that's the insult that wakes up and triggers your immune system to, or triggers, disrupts whatever, to then go on to this, the chronic illness. Yeah. So that's the grand hypothesis, but we don't, that's what we're chasing down. But essentially, at the root cause, I think, of that full-blown fatigue is likely to be a broken energy production. Yeah. It's not tiredness. The other horrific things about this illness is that you don't...
So I get what you're saying there. It's not tiredness like the people feel like, I haven't had enough sleep, I'm tired. It's like, I've just got no energy. There's no energy here.
And it's even worse than that. They have unrefreshing sleep. So they're so fatigued, and then they don't sleep well either. So it's kind of like the worst form of torture.
It's really interesting. I was talking to Dr. Jansen the other day about sleep cycles and things that happen in your brain where your brain all works in concert, and it seems to like it almost pumps and flushes the brain. So it's flushing out debris and toxins that you don't want, and it's pushing nutrients through. So if your whole energy production system isn't working, and that's not working real well, then your brain's not getting cleared or refreshed as well. You're fucked because you're just stuck in this cycle of not working right.
Exactly. And it's so torturous for these poor people with, like, they can be bedbound, and it's even in one study, it was listed as the worst quality of life of all illnesses. Because they don't die in any immediate way, it's like a state of zombification, right? Like where you're so ill and we can't do a test to show it. And so then the doctors think it's just psychological. So it's just horrific what goes on. So those are all the things we're trying to break down and say.
So, is there funding? Are you able to get funding to look into mitochondrial processes and see if that's it, and what impacts them and what can change them?
Yeah, so our study to date is crowdfunded because there's been no government funding to
how do we plug your thing? How do people tell us about it so people can go and donate?
Yeah, so our webpage, which was launched or co-founded by patients, so we're very patient focused. So it's always having the patient at the centre to understand, to work with the patient groups, and so the website is PVRA, so post-viral illness, but PV, post-viral research Aotearoa.nz. I mean, you can just Google me and Auckland University and generally one of those. PVRA.nz. Yeah, and so it's got, it was launched by the patients. I don't operate web page development, so it needed to come from not me. But essentially, that sort of shows you what we're trying to achieve. Anyone with long COVID or ME can also go to that page to download the participation form and sort of see what we're after. But yeah, the disappointing thing is there's been no urgent funding in this country. So as we're seeing come through now, a lot of countries are actually releasing things, sort of inquiries into the independent countries' responses. In some countries are specifically launching a long COVID inquiry, why didn't you act fast enough and things like that. So that's happening in Australia at the moment, a long COVID inquiry. There's another one going on in the UK. And the WHO are sort of at the centre of this, sort of saying we need to be doing more and all the rest of it. And so like from memory, I think there was 1.5 billion in US funding to go into long COVID initially. I think that was in 21. I can't remember exactly now. There was tens of millions in the UK. There's been 0 millions in New Zealand. So there's been no dedicated funding in this space. So our big question is, what is prevalence and all the rest of it, so that we can understand it better. We don't have that data here. So the short answer is we've been having to crowdfund because there's great urgency. There is massive urgency. These patients want answers now. There's been tons of literature come through. Like one of the things we hate hearing is, we don't know enough about long COVID. It's like, no, that just means you haven't read the literature. Like there is loads of research coming through. So we know that, things even like medical imaging, not mainstream medical imaging, because if there was, then, it would be, it would be clear paths to sort of diagnose this.
What is it? Because I've seen that term medical imaging. I don't quite understand that. What do you mean by that in that context? Like we could look at cells and see what's going on?
No, I'm just generalising things like MRIs.
Oh, okay.
Not my domain. Right. But, like as in things like to detect brain, problems in the brain or so on and so forth. And so there are particular groups that have done studies. I'm just trying to think of some of them. I think there was one showing, perfusion of oxygen in the lungs being impacted, whether it was in children or adults and so on and so forth. Like they're usually done in a research setting because they're asking the question, what's happened to this patient group?
And that's a tissue damage one. And I guess like people losing the sense of smell, that's nerve damage in the brain. We don't necessarily know enough. One of the confusing factors about impacts on the lungs, yes, Delta and prior was literally attacking the lungs, right? And therefore it would make sense that you get tissue damage. Since Omicron hit, people still get shortness of breath and all these weird symptoms. And in fact, the core groups of symptoms with long COVID is disruption to the nervous system. Dysautonomia is the term. And then neurological impacts, obviously, and then the immune system stuff. So that's kind of like sort of almost 3 categories. And if we hone in on the dysregulation, the dysautonomia or the nervous system impacts, what we understand about that is, we know what dysautonomia is in the sense that it's a disruption to our autopilot. And this was probably the most common symptom with people who don't recover. And it can even happen in people during their primary infection and then they experience it and then they recover. But essentially what that disruption is all the things we do naturally. Breathe, our heart beats, and our blood pressure, all those things that we don't, have to think about. Thankfully, right? Because that would be tragedy if we had to remember to breathe. So that system has been disrupted, and we don't exactly understand how, like whether that's
from the virus itself impacting our nervous system. But there's definite trigger points of the nervous system with this virus. So how that presents, very commonly tachycardia, heart racing. And then the medical term that you generally get diagnosed with if it's bad enough is POTS, which is, I always forget the full acronym, but basically what it means is when you're lying down flat and then stand upright, so post the medical term for standing up, orthostatic intolerance, there you go, post-orthostatic intolerance, yeah, there you go.
Syndrome.
So I forgot, S is syndrome, tachycardia, basically post standing up tachycardia syndrome. That'll do. So what, and people sort of are first frightened by this, right? If your heart's racing out of control, it's frightening. And we know that the virus can impact our heart. So it's always very important that people do go and see their doctors when they've got chest pain, shortness of breath, all those things. But once you've been cleared of any clear cardiac problem, it's generally...
It's a disruption of the nervous system.
Yes. But frustratingly, patients will often hear that there's nothing wrong. Sorry, you're just anxious. That's why your heart races. And it's like, oh my goodness. So what that is saying is that, and that's probably one of the most common, is POTS. So in a way to show this, is lay down flat and, you have your sort of your heart monitor. And some people are so sick they don't need to demonstrate it. But essentially, when you stand up and your heart rate goes through the roof, it's because you've just, your blood's now going to your feet and your body's not managing, the autopilot's broken. So the reason why I'm sort of tying this all into the lungs as well is shortness of breath could be that you need to do breathing training, so breath work. And it's not necessarily because you've got lung damage, it's your nervous system and
not
you need
telling
to
you to breathe
retrain
as rapidly as you should or need to.
So exactly, and chest pain could be linked into that. But as I say, this virus can be very serious in that it can cause tissue damage. That's why it's important to seek healthcare. But at the same time, that shortness of breath or lung problems can also be that this virus, people don't often realise that this virus can cause clotting blood. It triggers what's called immunothrombosis, which is a normal process for our immune systems to react to foreign objects. So it sort of, triggers clotty things, to basically capture bugs and go, hey, come over here, capture it in a big sticky goop so the immune system can deal with it. So we've known very early on that clotting is part of this. This is also why, some of the post-infection problems, irrespective of long COVID, is increased risks of heart attacks, increased risk of strokes. All of these things are linked to the fact that this virus can cause damage to our blood vessels and the clotty pathways. So it's just another one of those elements about this nasty virus that, the spike protein on the outside is a nice trigger of the clotty factors. So, shortness of breath could be linked to the fact that maybe your blood's not flowing like it used to because your body's still resolving. So, and it's not to say that this is dangerous in all of us, right? But we still don't know how to measure that trajectory. So that's why we sort of are very adamant and vehement about anyone who's looking for advice if they get infected and just feel that their recovery is a bit slow. If you're an athlete and a very fit person that wants to get back into your activities and you have any signs of shortness of breath or your heart rate racing too much, you're getting back to exercise too quickly. And even more so if you notice that you're fatigued after doing those things, again, it says you're not recovered and you need to rest. Like if there's one thing that athletes of the world who have not recovered are saying is just listen, because, you might become a little less fit for a while, but you may need that longer term to properly recover, because some people think it was their pushing too quickly that triggered their illness. And some of those people haven't recovered.
So again, it makes it sound like it could be mitochondria at the base of it. If you exert yourself and then you don't get energy back after that, because whatever you had, you've blown it. And if you haven't got if your cells haven't got the ability to produce energy through the mitochondria properly, you're going to feel completely knackered after you've burnt what you had.
So we don't know the direct link between, has SARS-CoV-2 caused the mitochondrial problems? Has SARS-CoV-2 woken up latent viruses? We don't know, right? But if you have any signs of those fatigue, and what that's also called is post-exertional malaise is basically the center or the core symptom of having these illnesses seriously. And that basically means any form of exertion. It's not just exercise. You know, if you're someone, you know, like you can even get post-exertional malaise from over-exerting mentally, going back to work too quickly, and emotional as well. So physical, emotional, mentally, all of those things can create a, you know, it can be 24 to 48 hours after your exertion. You're completely knackered. You're wiped out.
Yep.
And again, it's not just you're just a bit tired. You overdid it. Something's
you
metabolically.
You sleep. You still don't feel like you're getting any energy back, even if you have slept well, in that you've slept soundly, uninterrupted. You're just not getting that energy. But it makes total sense that it's mitochondria.
So our terms we like to sort of umbrella term is like immunometabolic as a classic one, because the immune system and something to do with the metabolic system has been disrupted in ways we don't yet understand, but it's immunometabolic. And at the core of this is some form of immune dysfunction, which is why, you know, why I'm trying to sink my teeth into all this. But there's so many, there's loads of, as I say, things coming through the literature. But, you know, we need to keep at it, like as in, it's not there.
Totally.
Diagnostics.
It'd be an amazing thing if you're able to pick out at least some of those causes and say, hey, it's this process in the metabolic system is being disrupted, and we can prove that, like, this virus, this protein is doing it. Yeah. Wow. That'd be huge.
Yeah, because we need treatments.
Yeah.
You know? And if there's anything that we feel most optimistic about is that we think that all the massive amount of research going on now is going to help all people with post-viral illnesses. And that's what we really want to be front and centre as well, that because we are listening to everyone. It's not about, long COVID's the hot new disease. It's post-infection illnesses essentially. Yeah. And I think that our immune system's at the core of it, and our past viral histories at the core of it. So the more that we understand about those and stop dismissing them as boring. Like, I can't even buy a test for my laboratory for tracking some of these things because they're, who cares? You know, everyone gets that virus. It's so understudied in some ways. You know, it's well studied in things like, you know, the link to cancer, like with EBV and how it can cause some cancers and so on and so forth. But just from a chronic illness perspective, that's
interesting too. Then, so like, what's with EBV? What is triggering those cancers? Because that sounds as though it's somehow disrupting the cell function. Because if they're normal cells and suddenly they start becoming cancerous because they've been exposed or they've been infected by the virus has got into those cells, and then they become cancerous.
Exactly. And so there's a whole list of EBV associated cancers. But again, remembering this is a tiny percent of humans, right?
So, of course, but still, just the mechanics of what could be going on there is really fascinating.
Yeah, and you know, and so arguably we're like, hey, what if EBV is the cause of all evil? Shouldn't we actually stop getting it? You know, and that's where I really that's why we really want to understand these viruses, because if the next phase of research is let's stop getting EBV, let's try and, you know, the best is to not get it. So, you know, like as in vaccinating to stop it in the first place, or is it going to be to come up with antivirals that target EBV?
Or it might be something that, maybe to stop getting it, or even it might be something to go, okay, we figured out what path it's dysregulated or disrupted. Fix that, turn that off, or turn that back on. Yeah, and effectively have a treatment for it, not a prophylaxis to avoid it, but a treatment for it. Yeah,
exactly. There's so much that we need to do in this space, and so that's the journey. And I mean, there's plenty of people with different hypotheses. You know, internationally, some groups are chasing down the fact that they think that because SARS-CoV-2 causes clottiness, that, you know, that the fact that we haven't resolved clotting, you know, like as in it could be sticky blood, if you like. Their hypothesis is that that's part of the drive. I'm more inclined to sort of say it's part of the picture.
Are they suggesting that sticky blood is then less efficient at transmitting oxygen and so
exactly.
you have an energy deficiency?
Yeah.
I guess that's...
So it's part of the hypothesis, but...
Does that stop? Does that sticky blood thing stop with people after a period of time? Because if it does, I guess it's pretty easy to prove that one and go, okay, it's now stopped and you feel better? Oh, well, that was your case then.
Yeah, that's a good question. And I think, I was talking to a collaborator over the weekend and he was sort of in that... he was sort of thinking that the severe form of that, or the detectable form of that, of the stickiness, does start to resolve, and you're still sick.
Yeah.
So, and that was my sort of gut feeling, if you like, when we first started knowing that this virus has the potential to be a bit clotty and sticky, make your blood sticky. Because, as I said, it's an immunothrombosis is an immune function to help clear infections. Mm. So, what I think's likely happening is that this happens to all of us, and then we naturally recover. We resolve that, but we just don't know how long for. So some of the risk factor papers that come out, sort of like saying for the next 6 months you're at high risk of all these things, that's probably what that's meaning. Like, we don't know how long we all wander around while our body's recovering. We feel normal, but are we back to equilibrium? Are we back to baseline? And so, it would be great if we had a tool to detect, oh, you're still a bit off, don't push your body yet. And obviously people do this themselves. They might be, as I say, the athlete that goes out and goes, no, I'm good. I've felt completely normal after that. Because, when we started publicising about not going back to exercise or pushing too soon, it was like, am I going to get long COVID? And it's like, no, it's not like a golden rule that everyone who exercises after COVID is going to get long COVID. It's more the fact, listen to your body. And this is one of those viruses. In fact, we should never push our bodies after an infection. Your body's telling you it's not ready yet. Yeah,
well, and again, I would say that with most viruses that I've had, I'm talking about good colds or the flu. I mean, the one time in the 90s that I had the flu, I was knackered. They exhaust your body. They for whatever's going on, your immune system, all the work it's doing, and then whatever else the cells might be doing, perhaps temporarily. Sorry, not the cells. The viruses might be doing temporarily to your body.
Yeah.
You feel exhausted.
Absolutely.
You don't get. It's not like, hey, I've got a flu. Let's go out and party. I feel awesome. It's like, no, you feel like you're just exhausted.
Yeah, absolutely.
So any sort of virus seems to do that to you.
Yeah, like when I got CMV, oh my God. Like, I was like, I've got a permanent hangover because, I was at the party age, if you like, and I'm like, this is horrific. And so what it told me was like, this is what, like, I was like, oh, is this feeling that feels like a hangover, does that mean a hangover is liver damage? Because when my liver was sick, it felt like the hangover. So I was kind of like, oh, that's what this correlation is. But it was so horrific. It was this wave of I felt so poisoned, and it was really odd. And this is kind of the closest I've come to some of these symptoms that people describe to me, because I felt like a wave would come over me, and then I'd be like, I can't move very quickly, and I'd feel like I can't remember if I thought I was in slow motion or everything was in slow motion, and it was such an odd feeling. And now I kind of understand that a bit when people are, when I see people with long COVID or ME.
Metabolic stuff.
Yeah, and it's kind of like, it's kind of involuntary. Like, it's bizarre. But,
when I fasted, when I did a 3-day fast, day 2 of that, I started to feel like that. Like, people were like, what was it like? Were you hungry? I was like, no, I wasn't hungry. Did you have real mental clarity? No, I didn't have real mental clarity. Everything felt like it was slowed down after about 2 days of not eating. Yeah. And again, I reckon that's because it was metabolic change. My body had no food. So my body's going, okay, maybe we back off on things a little bit in terms of energy production because we might need to conserve some. We're going into fasting crisis mode here, it looks like. But that's what I noticed. Everything just feels, and like I couldn't describe, like you said, if it was me that I felt slower or if everything else felt slower. It was one or the other though.
Yeah,
that was going on there. So what's cytometry?
Cytometry. That's my key tool of the trade. So cytometry means cytometry.
That's how I want to say it.
Because cyto means cell. Oh yeah. Metry, measuring. Yeah. So, and the word missing at the beginning there is flow cytometry. Cytometry means to measure cells, and then my tool of the trade is flow cytometry. So essentially, these are cool instruments that interrogate I was going to say cells, in my case cells, but basically particles in fluid or in a suspension by sucking them into an instrument under, so that's the flow part, fluidics, and then it goes past a laser, and the laser shines at the cell or the particle, and then it excites. Most of the times in the work that we do, there's fluorescent molecules on the cell surface, but essentially it's also exciting just a cell, like as in the light shines at the cell and it either bends around it or shines through it. So it gives us an indicator of the size of the cell. Right. But what we do, and especially in immunology, is we use fluorescent tags that stick to the cell surface. And all of those fluorescent tags can be excited by particular lasers, so basically a whole rainbow of different colours. And why do we want to do this? So that we can unpack and see all the different immune cells in the blood, for example, that are present. And we start to ask questions, so we can answer loads of questions with flow cytometry based on looking for the cell surface molecules or even function of the cell. There's tons of things you can do with it, but flow cytometry is the tool of my trade. So I spent a large portion of my career after my PhD sinking my teeth into this technology. I love it. It's super cool. So do you use it for things like looking at someone's blood? Is that what you use? Yes.
And then is it to say, oh, hey, we should be seeing helper T cells, and we don't see any helper T cells here. So that's exactly a problem, or that's something interesting here.
And in fact, good example, because this tool was probably most famously used. I described earlier about HIV, and that when the helper cells fell away, it started to indicate you are going to progress to an immunocompromised state. And this was the technique that was used. So on real basic terms, we would count how many T cells are there, and then we would look at the ratio between how many helpers are there and how many killers are there. When the helper T cells dropped below a particular ratio, it meant your immune system was starting to be dysregulated and that you were at more risk of going on
into AIDS.
Yes. Yeah. And so there was a specific ratio that then would give you the AIDS diagnosis. Yeah. Wow. So and we're now looking, through my career, it's been so exciting because when I started using flow cytometry for my PhD, we were looking at the way we describe it as colours. Think of colours as the fluorescent molecules. Right. We were looking at 4 fluorescent colours. And quite sort of mainstream, easy to do. It was always in the back of an immunology lab. Anyone could rock up and check, the 4 different fluorescent colours. And then over the years, it's always like, how many colours can you measure and what's your machine can do? And so there was massive pressure on the industry, both in developing new fluorescent molecules so that we could detect the differences between them all.
Detect more cell types.
More cell types, exactly. And also the instrumentation, we needed technology advancements so that, we could look for more cell types, exactly. And so just to describe that process a bit more, when we have a cell, it's got lots of proteins on the cell surface, right? So when I talk about fluorescent tags, the fluorescent tag is conjugated or, bound to, we buy these specialized reagents, but basically they're highly specific, again, so they will only stick to a particular molecule on a particular cell. Right. And that's how we get that specificity. So we might have, for example, there's a molecule called CD4. People hate immunologists that just go CD this, CD that, because it's just talking in code, which is why I always call them helpers and killers. But essentially a CD4 T cell has a molecule, all T cells have CD3 on them. So we stick, one of the molecules will glow because all of the cells...
For CD3, so these are all T cells.
Yep, and we can say they're all green. And then we'd use a blue colour and a red colour. The blue would be on the killers and the red would be on the helpers. And that's how we start to dissect out the colours in a big spectrum, a big rainbow spectrum of different colours.
And then I guess, does the machine do the counting for you? Like it flows through at a certain rate and it'll say, it picks up for you 25,000 T killers and 6,000 of these, and that ratio is okay. Well, you figure out the ratio, but it just does the counting for you. Obviously.
Yeah, exactly. So what it's doing is interrogating cells one by one. So, we often will look at 1 million cells coming through from a single blood sample, and we will look to see how many T cells you have, how many B cells, and all the different types of cell types in your bloodstream. And then we can also use some of these labels to detect function of those cells. What we often look at in the context of viral infections and things, depending on what the question is. But we can go looking for what we call exhausted T cells. And they literally mean what it sounds like. And we try and find if with some viral infections, it might exhaust your immune system. And that might mean, how I described that you make a big army.
Yeah.
The army dies off, but they might be constantly stimulated because for whatever reason, the antigen is still around. So like it could be that, it took ages to clear a virus. And so you create constant stimulation of that T cell. And it's like, I can't keep going because, they have a massive ability to clone, like make copies of itself. But at some point it's going to run out. It's not infinite. And so we have markers that can detect how exhausted a T cell is. And so, so that means they're less fit, literally.
To do their work.
So if they see that infection again, they might be like, oh, God. Yeah. And then, the infection might kill them. As in they might go, oh, I'll kill you, but then I'm dead. So point being is there's lots of cell surface receptors or molecules to understand function. And it's all using this instrument we call a cytometer.
Can you look for viruses with it, or do you look just for human cells?
Viruses are too teeny.
Right.
So, but we look for indicators of viral infection. So what we do look for is we look for viruses that have cells that have responded to viruses.
Okay.
So we can look for those with bespoke reagents as well. So say, for example, you've just had an infection and your army's still, floating around, as in they're not all died off yet. You can use molecules that pick up that receptor on the cell surface with a fluorescent tag. And so you can say, oh, look, this person's got, has had SARS-CoV-2. This person's just had EBV. This person's just had CMV, because there's loads of them detectable floating around the blood.
And maybe you see like a large number of T cells. So not only can you see that there are cells with the indicator, but also what stage they're at.
Exactly.
They're exhausted, or the response is finished, or the response is probably still on the way up because the numbers are growing.
Exactly. Loads of things. Because clinically, this is research stuff, right? But clinically, if you're unwell for whatever reason and you go along to the doctor and you get told you've got a high white blood count or something like that, often your sample will then go to a clinical cytometrist and they will be looking to see what cell type you've got too many of. And it could be because something's grown and that's sort of the process for blood cancers, what cell's grown out of control, and then they start to do the same thing in a clinical way.
Wow.
Yeah. It's a super cool tool.
Yeah.
Yeah.
It sounds pretty cool. It sounds pretty interesting. Yeah.
So, and as I say, it's normally for cells, but we can look at bacteria. Basically,
bacteria are big enough, are they?
Yeah.
Yeah.
It gets more tricky because they were developed for cells. So bacteria are much smaller, but the smaller things get, the trickier it becomes. But
I guess the technology improves over time as well with the ability to do that.
Yeah.
And do they start to put load onto these machines functions like I was saying, yes, they can count. Can you program, hey, count the cell types and tell me the ratio so I don't have to work it out?
Yeah, you can do all that sort of stuff. Yeah. And the other cool thing is, so we have two types of instruments. One we call an analyzer, so it just literally analyzes them and then goes to waste. But we have the same technology that sorts the cells. So say, for example, So say, for example, we were talking before about HH36 impacting the helper cells. I can put my sample through a cell sorter and fluorescently tag all the helpers and sort them into a tube. So I just go looking at my helpers.
Go extract them and then use them.
Yeah.
Wow.
So we do that also, if we want to just study T cells, we tag them up with their T cell only molecule. We put them through the instrument. We say just collect them and we squirt them into a tube.
Do you know how does it collect? How does it actually push them? Does it push them with a laser or how? It's
all quite cool and physicsy. Yeah. Essentially, so it's the same software that we're used to looking at, when we're analysing cells. So we literally call them gates. We draw a circle around them and say, those are T cells. Right. And then all the physics happens. And so, as I said, our samples get sucked into the instrument. Yeah. They need to be in single file.
Really?
All the fluidics are you've got to go one at a time.
Wow.
So, it's often a very, very fine bore fluidic system so that we push them on purpose into single file.
And then one at a time, you can just flick it over here, flick it over there.
Exactly. So as it wanders past the laser, the laser hits it.
Identifies it.
And it's all super fast and everything. It identifies it's there. And then the software tells it, I'm interested in that. So as it comes down a bit lower, it gets, and it's basically in a salt solution. So it's like salt water, saline. And, you can then charge
a particle and pull it in if you want to.
Exactly. So what happens is, in the cool physics of these instruments, every cell gets encapsulated in a droplet of saline. So the cell is not impacted by, the laser getting zapped or anything. It's a ball of saline solution with one single cell in there, and you charge it literally with an electrode plate, and it goes zip, and it spits it into the right tube. It's incredible.
That's pretty insane technology to be able to do that sort of thing. Yeah. So
part of my PhD, the cool thing that I was doing a lot of, we were cloning T cells. So you do that. You have a 96 well plate, and it's got all its juices ready and waiting, and you spit single T cells, one all by itself, lonely T cell, into each well, and they don't have a high success rate because you're spitting them into a lonely well, and we have sort of a feeder population that dies, but essentially the upshot is that we're triggering them to clone themselves so that they're an absolute replica. So if I did that, say for EBV, I can make a whole plate full of cells that can kill EBV infected cells. And so it's super cool doing things like that, where you make a clone.
So when they clone, they're in there. You put some proteins or something around them to trigger the process, and then do they do is this like a normal cell? Is it meiosis? Is it called, where they sort of mitosis? Yes, is that it? Yeah,
exactly. So they stimulate to say divide and grow, and we're giving them all the right nutrients and signals to do that. And literally, yes, they divide in half, and they keep doing that. We have this super cool dye. Again, I did this in my PhD as well, where we stain them green, and when a cell divides, the dye halves, and they keep doing that. So you can actually count how many cell divisions it's been through by it's losing the intensity of the green dye. Super cool.
Scientists, you're clever, you bunch. That's cool.
It's fun stuff. Yeah. So that's all the sort of tools I was playing with during my PhD to sort of really understand, and these are, human cells out the body, they don't live forever. So it was all about harnessing them and giving the right proteins to tell them to grow and divide, but not exhaust them or make them, go to the end of their livable life, if you like.
Do you know, how have I asked this the right way? Do you know why they would die? What's it called when they're out of the body? Is that when you're
ex vivo?
Ex vivo.
It's ex vivo or in vitro. Yeah, so ex vivo comes out the body, and then we grow them in vitro in the lab.
When you're growing them in vitro in the lab and they were dying, is it because you weren't feeding them the right nutrients, or was it because they were somehow figuring out something's not wrong with my environment and they were apoptosis?
Yeah. It could be both of those things. Exactly. So my job was to sort of see how much how far will they grow, because they will. In fact, those were the questions. Like, if I give them a signal at the beginning, how long do they keep growing for
and keep cloning for?
Yeah. And do they slow down, and therefore do I need to give them a new signal to make them do it again? And how long can I keep doing that for? And like one of the phenomenon that I was playing with is that what we call activation-induced cell death, where you may have gone through one round of stimulation and they're sort of sitting there ready to go. But if they weren't in the right environment, and we said, right, here's your trigger, because we stimulate their receptors, mimicking a response. If they weren't in the right protein environment, they'd be like, activation-induced cell death, like as in, because it's mimicking in the body overstimulation, and you can't blow up, infinitely with T cells, right? Your body will be completely controlling how many of those cells can be around, so you don't fill up with cells.
So out of interest, did you ever have to put helper cells in with those T cells to try to help get the response you want, or to get them to grow longer? Or
that's a very good question. The cells that I was manipulating were the T helpers, but when we clone them, yes, what we do is we use a cell line, which generally just means we've immortalized them, as in it produces, immune molecules to keep cells happy, because they like to touch, be in a cozy environment as well. And what we would do is irradiate that what do they call it? Supporter cells, essentially. That's called. So you give them a a toxic signal like irradiation, so that they're going to be alive long enough, but so they don't take over. So so they're going to sit there and produce molecules. So that's for cloning. And then it would produce enough help and signals to allow the clone to grow. But even when we get straight T cells and grow them in the lab, we, just like you sort of referred to, do we need to provide help? Literally, yes. What we're doing is we provide the cytokines or chemokines or molecules that a helper cell normally produces. So, we would, when we strip cells out of their natural environment, we always have to then come up with what we have to add back in to keep them happy. Because for example, when we take our white blood cells from our blood and we create, we pop them into the laboratory setting, we call that PBMC, so peripheral blood mononuclear cells, all the white blood cells into the lab. And they can sit quite happily without any external triggers because they're all used to hanging out together.
Right.
But if you then isolate out, say, naive T cells and put them in a well, they'll be lonely. They might not have the natural signals. And those are all the things that we try and dissect out because we don't naturally know what the key signals are that they might need. So that's what we do.
It reminds me, I was talking to Bronwyn Connor the other day, and she was telling me how they're using 3D printed biogels to allow them to, when they turn skin cells into neural stem cells, to give them a foundation in which to grow, so they're not just growing in a two-dimensional plane, because she feels that if they grow in a two-dimensional plane, the results they get aren't accurate, because in vitro in the body they are growing in 3 dimensions, and so so that we're always trying to replicate
mimic, exactly. And that comes down to sometimes we want to stimulate cells in the best way without being like sledgehammery, or we want to create an environment. So 3D culturing is becoming more and more common, because absolutely, for example, when I just described the PBMC, when we put that into tissue culture plates, they're like, ooh, plastic.
Yeah.
Ooh, what's this? So they stick down. Like, they're not normally going to stick down. So when a cell goes, ooh, plastic, and starts to stick to it, it's naturally going to start releasing molecules to do with why it wants to do that.
Yeah.
It thinks it's now becoming a macrophage, and I'm going to now form, a tissue or whatever it's
going to do. It
starts spitting out different molecules. And so the more that we can mimic in vivo situations, the better it is, depending on what the question is we're asking. But certainly, there's so much pressure now to try and mimic as much as possible, so that, we can better understand what happens in the body.
Get better results.
Yeah, totally.
Wow. Hey, thank you so much. It's been really, really interesting.
No problem. It's always fun to talk about.
Well,
so many different things.
It's really good fun to be able to sit down with someone and just talk for a couple of hours. To actually chat, and particularly on a topic like this, which I don't know anything really about, but I am interested in. It's really nice to be able to. And
the short of it is we don't know anything about immunology. That's
complex.
Kind of where we're still at.
Yeah.
It's we need to get rid of allergy, autoimmunity, cancer. All of these things is where our immune system slightly breaks down. So therefore, we've got so much more we need to know.
Sharks, aren't they good for not having cancer for some reason?
Yeah, I think there is that thing, eh?
Yeah, that to me has always been interesting in that field. It's like, okay, well, let's go look at sharks. Why don't they get cancer? What's going on that's different with them?
It's hard to know exactly what all these things are. I mean, we manipulate our bodies too much as well, don't we? Like, as you say, we eat processed foods.
Yeah.
Or,
well, also, modern life, all sorts of things.
Yeah, exactly. If we stripped away everything and went back, hundreds of years, we just had shorter lifespans and all the rest of it. So.
Well, and that's the other thing too. We start to deal with issues that we probably wouldn't have had 400 years ago, because we would have died at 40, or would have been a good life 60 if you're really old.
Yeah.
Now my nan died at 102.
Wow.
People, I reckon the next couple of years, I would say the next couple of years, let's say the next 10 years, people will be adding 10 or 15 years on their lifespan, I think.
Maybe, but we've just had a pandemic. We're still coming through it.
True.
And there's a chance that that's going to impact our lifespans.
I mean, generally across the board. Yeah, but I mean, specifically, like some people will be able to go I've talked about this with a couple of people. It's like, we could in the future 3D print a heart. If we know you're going to have heart problems, let's just 3D print you a new one out of your cells, which you took from your skin. So it's a really good mix, doesn't get rejected. Like, Okay, that's pretty cool.
Yeah.
Age 80, you got a new heart. So instead of dying of a heart attack at age 92, you might just keep rocking on. Now you've got to deal with something else. Now it's some neurological thing we've got to deal with. Get rid of your Parkinson's. Well, we can do that too. That's all right. Next, give it 20 years, I think, lifespans.
Yeah, I mean, we're learning more and more all the time.
Yeah.
And it's just crazy when we think of some of the things that feel like we're only just starting to scratch the surface. Like, as I said, I think microbiome we know lots more about, but I think there's a ton that we need to still understand. Yeah,
well, because someone was telling me, I don't know what the number was, some part of our body, some huge part of it is other cells.
Yes, we've got more, like there's less human cells than there are other species. Basically our microbes, because we've got all sorts of species everywhere, and I think the statistic is that there's more cells of other things than human cells.
Yeah.
Yes. That's why I said. That's why I call ourselves apartment blocks.
Yeah.
We're just carrying them around.
Yeah.
It's hilarious that and we've got to keep them happy, and so and they can get out of whack so easily.
It's so amazing that we actually live as long as we do with so many complex systems and things that, when they go wrong, are problematic, and yet in most people for 80 years, they don't go wrong.
Yeah.
That's crazy.
And there's a lot of these species are hijacking us, and I think, the futuristic thing will be understanding which one should we invite in and foster and get rid of the ones that we don't want.
And which ones do we leverage? Like which viruses do we take and say, hey, we want you, but we're going to tweak you so that what you will do is become our mitochondria protector, because we know you love to get into the mitochondria, but you're going to do this to stop the other ones from causing a problem there. Because
and that's the thing. Like, there must be so many of our microbes that live in us that we don't yet know their symbiotic relationship with us. Yeah. They're in us so that, they live.
Yeah.
And, what process are they making us, allows us to thrive?
Well, that's the thing. I guess the ones that have stuck around through evolution are the ones who do give us some benefit as well. Yeah. It is symbiotic.
Totally. Exactly. Well, I mean, that's what our microbiome is a symbiosis. It's just when the bad guys get in, it's harder to get them out. And we're still understanding about those things, I guess, is how to get the bad guys out and how to rebalance that. What's the fastest way to do that? That's where the fecal transplants come in as well. And I think in the future could be more of that, sort of like, yes, your disease might be this, but let's start with your microbiome, so that when we treat the other thing, you're more likely to
you got a good
bounce
base.
Back. Exactly.
Yeah.
Exactly. So, it's so complex and all overlapping, but we know that, the molecules that our other species living in us give off, can be positive or negative, and we need to understand more about that. And we absolutely know that that's what's feeding into the immune system, and, and then how it impacts our brains and all the rest of it. Yeah. I think one of the frightening things, if you like, about the SARS-CoV-2 virus is how many people were neurologically impacted and go, oh my goodness, it was so bizarre how much it affects my thinking and all the rest of it.
The brain fog and I said, loss of smell, and those sort of things.
Yeah, and that could be impacts on the nervous system or direct organ impacts. Again, like, yes, there's been incidents where people have found SARS-CoV-2, the virus, in brain tissue, but again, it's not a hard and fast rule that, like the general feel is that we're getting immune, basically molecules impacting our neurofunction, not the virus in there. But the virus can still get places.
Yeah, virus is causing inflammation in your body, and some of that's signaling to parts of your brain, so you get an impact from that.
Exactly.
It's all connected.
Exactly. Yeah. So it's pretty intense, like sort of thinking, these little viruses have the ability to sort of impact our brains. Because one of the things I was saying early on was toxoplasmosis. Creepy little thing, that creepy little parasite. And, it's incredible how that evolves, how that replicates and continues, is that it makes a mouse go, I don't care about cats.
Makes men less risk averse and women more promiscuous.
Well, there you go.
I got it about 10 years ago, and the reason I know that I got it is because I got really sick with it. I thought that I had like lymphoma. I thought I was getting cancer.
Did you get a It's swollen
back of
lymph
my
nodes?
neck. Yeah, back of my neck, like up here.
Yeah.
And up here for, I don't know, a week or so. I couldn't get to sleep. I had such chronic pain. I was like, there's something wrong with me. And Renee said, go to the doctor. I went to the doctor, and the end up diagnosis that came back was, you've got toxoplasmosis. And so now this is, I don't know if this part is psychosomatic. So I went and researched, like, I'll Dr. Google what, okay, wow, this is a really interesting thing to have. Lots of people have got it. Most people don't get sick with it. You just get it. You don't know. One of the things that was meant to happen, which I noticed, was when you get it, you have a dip in your reflexes for a period of time, and then it comes back. Sure enough, I reckon that when I was driving in the couple of weeks afterwards, I felt as though I was less capable driving, like more likely to run up the back of a car or find myself approaching too closely, like, hey, this isn't like me when I drive, which I
was trying to knock you off.
Well, yeah, maybe it was. but it made me think, hey, maybe that is that lack of reflexes that's going on for a period of time. Yeah.
Because I've had toxo as well. I'm a walking viral bag and parasites, all the rest of it.
We all are.
Yeah. And my story was that, I had a subclavicular lump. So I had a raised lump on my clavicle. And I'm like, and I only noticed it was something like I was laying on the couch and, I could feel something tender, and then I'm like, this isn't right. And I was told I was a hypochondriac, oh, whatever, everyone's got lumps and bumps, that's fine. And then I was talking to a colleague and she was like, get to the doctor. So I went to the doctor and, felt this squishy lump, sent me off for blood tests, and then said, I'm going to say that word you don't want to hear. I'm going to say the C word. And I'm like, oh, God, things are escalating. But I'd already sort of was comfortable with the fact that there was a link to subclavicular swollen lymph nodes and toxoplasmosis. Because I think the first appointment I had with her was, to go and get blood tests. And clearly she thought, test for toxo. And, your results come back that you've at least had it recently because of the immune response. And I'm like, oh, it's totally this. This is exactly what this is. And and then so I started looking into it, and I was just like, this parasite's such a creepy little thing, right? But then that's it, so when I went, I had to have a ultrasound, and and I could understand what the the doctor speak was, and it was kind of like it wasn't vascularized, and I was kind of like, this is clearly not a tumor of any sort. It was a swollen lymph node, because your immune system basically wraps around the creepy little thing, encapsulates it to sort of shut it down. And so it was an inflamed lymph node that then eventually resolved. But it is incredible. The life cycle of toxoplasmosis is that, people often say you catch it from cats, and it's usually from, contamination because they, poop it out and all the rest of it. And that's how it's sort of quite commonly picked up by children and all the rest of it. I had a cat, but I always laugh sort of saying, I didn't get it off my cat. He hates me kind of thing. Like, there's no way I got it off my cat. Because at the time, I think one of my rationales, how I think I got it was from eating a lot of game meat, because it can be in
red meat.
Yeah. And, but anyway, however I got it, when I was researching it, I was just like, this is so creepy. So the life cycle has to be complete. I can't remember which way around. It completes its life cycle in a cat.
Yeah.
And so if a mouse is infected, it can no longer, I think, is it smell cat urine?
Or it's not. It's no longer scared.
It's not fearful of cats.
No longer fearful of cat urine.
But I can't remember if it's to do with the fact that it's. And so it just goes, no, I don't care, come get me. And so then the cat kills the mouse and gets
the toxo gets into its gut where it reproduces.
Cool. And so there's this whole risk averse thing. Yeah. And so, we were having a bit of a laugh that, what if, SARS-CoV-2 is impacting humans, to go, it's fine. Because we were laughing at the sort of the phase where, there was this tendency that once people had been infected, they were like, it was fine, come on in, the water's boiling, sort of like, and we were like, oh my God, maybe this virus is impacting humans as well, to go, it's fine, everyone come get infected because it's great, it's no problem.
If you're a virus, that's what you want to do. I mean, you're trying to express your genes. If you can convince people to happily express the genes and share it, it makes great evolutionary sense for a virus to do that.
Yeah. And toxo's gross, but it was quite funny that I wasn't. I thought that was me sort of going, oh, this is my cool little analogy, but others had said it too.
Yeah.
Yeah.
But toxo, though, ends up forming plaques in the brain, which I don't think you can shift. Goes dormant. Or I don't think you ever lose it. It remains in the brain.
Yeah. Because it's another link to, becoming immunocompromised. Like, so again, like with HIV, if you've got toxo, if you get toxo, then you're less likely to shut it down and all the rest of it.
Right.
Yeah. So we hope it doesn't end up in the brain.
It's got some weird things too. I'm pretty sure there was a study suggesting that there's a link between toxo and soccer players. Like on a country, it was
Soccer players?
Some correlation between the level of toxo in a country and the number of professional footballers they produce.
What, it makes you a good soccer player? Is that what you're saying?
Yeah. Well, maybe I don't know. Maybe the risk-averse part of it makes you take more chances playing soccer, and you a good soccer player. I don't know. I'm sure it wasn't like causal determination, but there was some sort of correlation. Oh, lots of good soccer players, lots of toxo. Interesting.
Because I know this, at least when I got it, which was quite a while ago now, but there was a group at Auckland University studying acute infection, because it's really hard to study acute infection because you don't necessarily know you've got it. And I was like, oh, this is interesting. I can't remember who the group were, but there was an acute study going on. Yeah,
it'd be interesting to know more about what they learn about it.
Because it's obviously really dangerous for pregnant women.
That's right. I don't know why. Does it cause abortion or something, doesn't it?
It's definitely problematic to babies in utero, yeah. So that's why it's always, super careful about pregnant women. Because I think I remember during my Googling on it, saw some actress that had contracted toxo whilst pregnant, but her baby was fine. I can't remember who that was, but yeah. But that's a parasite. It's such gross things.
Creepy.
I know. Creepy little creatures.
I read tapeworms. Tapeworms can live for 30 years.
Yuck.
Like, who's got a tapeworm in the gut for 30 years?
Well, sometimes there's those people that want to have worms for weight loss problems.
They used to do that, didn't they? The Victorian era? Have the tapeworm egg? Yeah. They look much better now.
Then you could feed your worm. Yuck.
All right, that's it. Oh, sorry, let's wind this up. Thank you so much.
You're talking disgusting, creepy things.
We can come back into another one on parasites. Oh, that was fun.
Cool.
Thank you.
No worries.
Did you have fun?
Yes.
Good.